All right, so before I get started, I just want to acknowledge all of the talented people in both of our groups who've actually done the work that I'm going to talk about today. And I will try to remember to mention their specific contributions as I go through the talk. And I also just want to echo the sentiment that several other people have already expressed, that this has been a great experience and it's really been a pleasure working with and interacting with all of the other groups associated with FaceBase, and especially my collaborator, Matthew Harris. As you can appreciate by looking at this top-down picture of a zebrafish skull, the overall structure in the adult is largely similar to humans and other model organisms such as mouse and chick, but many of the structures that are most relevant to human craniofacial defects, including the bones on the top of the skull, develop well past the early stages that have been most intensively studied in zebrafish. And I think actually the last talk and the modeling that Eric has been doing of some of the human craniofacial disorders illustrates both the strength that some of those approaches can bring and also some of the limitations. And when we first conceived of this project and began working on it, we were building on a really important foundation in the zebrafish related to early craniofacial development. And this foundation included many detailed descriptions of structures and gene expression in the larva, and many genetic screens that yielded mutations that affected larval development. And however, much of this knowledge was of somewhat limited usefulness in explaining the mechanism of many human craniofacial disorders. And by the very fact that many of these disorders manifest in live births, we know that they're having their effect somewhat later in development. So partly for this reason, the mouse and chick have historically been more widely used as models for human craniofacial development. And both of these systems also have rich and detailed descriptions of development and adult anatomy. And in the mouse in particular, very sophisticated tools for genomic engineering. So when we first began our work, the Kimmel Lab had created FishFace, and this is a community resource describing early aspects of zebrafish craniofacial development. And this data set, which is still available on the FaceBase site, is made up of really beautiful, detailed images, mostly from confocal microscopy, and it served as a valuable resource for many groups, including our own. And we were inspired by this example in the planning stages of our own work, and we wanted to create something equally useful for later stages of zebrafish craniofacial development. And as useful as this data set has been, it did suffer from two major limitations. One is that the data was derived from fixed samples, so it's very difficult to capture a lot of the dynamics of craniofacial development. And it was also limited in the time points. It covered stages up to about 3 weeks of development, and then also had a description of adult anatomy. And we really wanted to fill in some of those missing time points in our work. So we saw in the gap between the zebrafish, what's been described in the human, and what was known from the mouse and the chick, in the gap between these bases of knowledge, we saw an opportunity to connect them and capitalize on the strength of all of the approaches. And we set out to make zebrafish an even more powerful model system for human disease, in particular congenital craniofacial disorders. So in the course of working on our FaceBase project, we've developed tools to better visualize and describe later skull development and anatomy, bridging the gap between the larva and the adult. And this has enabled us to describe mutant phenotypes more quantitatively and facilitated comparisons with human and with other model systems. And we've also worked to create an anatomical atlas that will be accessible to the community and will help other groups analyze their mutant phenotypes and also draw parallels with these other systems. So in my lab, we have exploited some of the primary strengths of zebrafish as a model system. Its accessibility for imaging throughout development, and the existence of many fluorescent transgenic lines that label specific cell types and tissues. So our imaging has relied on low magnification confocal microscopy, and we've largely used these two transgenic lines that mark respectively cartilage and bone throughout growth and into the adult. Because we're imaging live fish, we can watch the dynamic processes of skull formation as illustrated here. At this point, I want to stop and make a comment about developmental staging. So early larval development is not extremely variable and can be described fairly precisely by talking about days post-fertilization. However, at these later stages, the growth and the achievement of developmental milestones becomes much more dependent on growth conditions and is much more variable. So to compensate for this, we use standard length to calibrate all of our imaging data. And so from now on, I'll largely only refer to standard length. Although I usually also have the days post-fertilization on the slide as a reference point. So these are images of cartilage and bone formation in one individual fish over a period of 2 weeks, imaged successively. And this ability to follow skull formation in live animals during a time when other model organisms are largely inaccessible has provided us really important insights into the process. So one important example is the way in which we've been able to observe the earliest stages of frontal bone initiation, seen here in an individual fish. And it starts off with only a handful of cells, and these first appear in close association with a peripheral cartilage element. And over a period of several days, as you can see here, the cells increase in number, but still remain tightly associated with the cartilage. So through similar observations on a large number of fish, we've developed a model of important stages in growth of the frontal bones. And we can use this as a framework to think about both the anatomical events that are taking place and the mutant phenotypes that we're observing. So in the first phase, when the cells remain closely associated with the cartilage, the area of the frontal bones does not really change much as a function of standard length. And we've basically, for all of these measurements, taken the images that you saw in the last couple slides and created a projection of the Z-stack of the Z-stack from the confocal imaging of just the bone expression. And then in Fiji, we've measured the area of those frontal bones as a function of the standard length of the fish. So you can see in this first initiation phase, when the cells are first appearing, the area of the bone does not really increase much as a function of standard length. In the second phase, the bones enter a stage of rapid planar growth, when the two frontal bones, and with a slight lag of several days, the two parietal bones grow towards the apex of the skull to cover the surface of the brain. And then finally, in the third phase, the bones make contact and begin to form overlapping sutures, at which point their growth is measurably slower. And we can quantify that growth rate also in the third phase of suture formation. So to illustrate part of the utility of these measurements, I wanted to just briefly show comparable data from two mutants. The first of these is Chihuahua, and this is a dominant mutation in a type 1 collagen gene, and it's a model for human osteogenesis imperfecta. And the second model, or the second of these mutants, is called Toth, and this is in an unidentified gene that also broadly affects growth of the skull and the skeleton. And superficially, the skull growth of these two mutants looks similar. They both display significant delays in growth of the bones relative to their siblings. However, when we plot the growth data, it's apparent that the two mutants show a very different pattern. While the Chihuahua mutants show a delay in frontal bone growth relative to standard length, the Toth mutants actually show the opposite pattern of growth, with faster frontal bone growth relative to standard length. So in addition to the standard markers that we've been using for most of our imaging, we've also been documenting the expression of other transgenes of potential interest to the broader community. And so I'm showing here two examples of transgenes based on enhancer sequences associated with human disease genes. In one case, the RUNX2 transcription factor, and in another case, the BMP-ER gene that encodes extracellular modulator of BMP signaling. And both of these enhancers are active in precursor cells at the edges of the growing frontal bones. Another important example is this transgene, which is based on regulatory sequences from the CTSK gene. And this was generated by Joanna Lopez in the Harris lab. And this transgene serves as a marker for active osteoclasts. And Kelly in my group has been working to generate an atlas of that activity during these stages of craniofacial development. So as you can see here, the cells are sparse and scattered, and their positions vary both from one fish to another. This is showing two different individuals at the same stage. And also across time within individual fish. So this data is by nature a little bit different from some of the other confocal data that we've been depositing on the FaceBase site. And so we had to come up with a little bit more meaningful way to represent the data. And so to effectively represent this, we've overlaid the data from several fish, and this is shown here in dorsal view at two different stages. And so we're making the data, or in most cases have already made the data from these constructs available on FaceBase, and the transgenic lines themselves are also available, and we've already shared them with many other groups. And we hope that these the combination of the data and the available lines will serve as an important resource for the community. So as important as the live imaging has proven, it also has significant technical limitations. And first, because it relies on confocal imaging of the transgenic lines, it's limited to earlier developmental stages. It's also more time-consuming, and repeated imaging is pretty stressful to the fish. So this limits the number of individuals and the amount of data that we can collect. So the Harris lab has been relying on high-resolution micro-computed tomography, or micro-CT, to generate images of wild-type anatomy in later juvenile fish and in adults. And this imaging modality captures data with equally high resolution in all axes, and this really lends itself to three-dimensional reconstructions. Additionally, the ease of micro-CT imaging with no requirement for transgenes in the background or for special tissue preparation makes it an ideal screening tool for more subtle abnormalities that might otherwise go unnoticed.
And you're at 5 minutes.
Okay, great. Thanks. So this slide is a beautiful illustration of that, showing many of the mutants that the Harris Lab has scanned, and the data for most of these, or maybe for all of these at this point, has been made available on FaceBase. And importantly, many of these are mutants in genes or in pathways that are affected in human craniofacial disorders. And that's reflected in many cases by similar anatomical defects. So I'm sure you can appreciate from the above images the complexity of the anatomy in the adult skull. And there are some really nice published descriptions of the formation of all of these dozens of bones. But we wanted to create a more accessible resource to aid in visualization of the complex three-dimensional relationships. And we were inspired by the reconstructions that were published by Yang's group based on micro-CTs of the mouse skulls during development. So using their approach as a starting point, we've created similar reconstructions both for development and also the adult skull based on micro-CTs. So the first models were created by Osman in my lab based on the confocal imaging of cartilage and bone. So he segmented all the structures from the confocal data and then assigned colors and annotated them. And he then exported them into Acrobat and created interactive 3D PDFs. So each annotated structure is associated with a color and a label, and as you can see here, the entire image can be freely rotated, and the interactive buttons allow for each element to either be made translucent or to be removed entirely from the image. And the PDFs are files of modest size. They can be easily downloaded and viewed with Acrobat Reader. So this is all great when the fish is small and the anatomy is relatively simple, but the real challenge for us was to create a similar atlas of the adult anatomy. And so starting with micro-CT data from a wild-type adult fish, Catherine in the Harris Lab has similarly segmented the data, and Osman has generated corresponding PDFs. And as with the confocal models, the image can be freely rotated, and because of the complexity of the anatomy, we grouped related bones together. So entire group can be controlled with a single button, or individual bones can be controlled separately. And as with the earlier models, these models of the adult anatomy will be available on FaceBase for viewing or for download. So I'm just going to summarize really quickly what we think we've accomplished to make zebrafish a more effective model for human craniofacial disorders. And I'll stop here and leave a couple minutes for questions if there are any.
That's great, thank you. We do have a question in the chat from Bhavna Tandon. Are the embryos fish treated with PTU or have lack of pigment due to some other mutation?
No, so all of our confocal imaging was done on fish that were wild type for pigmentation. So we thought initially we might have to use something like that to enable the confocal imaging, but it turned out to not be a problem.
Okay, Timothy Cox, there are numerous groups developing automated segmentation tools. Are there plans to exploit these?
Yeah, that's an interesting question. So we, our approach, I would say, was kind of semi-automated, but largely relied on manual double checking of the, so in other words, we would set thresholds to automatically segment the data, but we found that Certainly for the confocal data, it required a fair amount of manual cleaning up. So we didn't do the segmentation for the confocal or for the micro CT data. So Matt might be able to answer a little better with respect to that data. I don't know. Do you have anything to add?
So the adult skull is far too complex to let the current automated systems do segmentation. So that we really had to go and make sure by hand that bones that we knew were anatomically distinct were being separated out. I think over time and with the increase in sensitivity of different micro CT systems that are out there, it'll get to that point. But what we wanted is to really get a working model out to the community so that you can actually leverage analogous systems between zebrafish and mouse and ask questions about gene function.